we finished basically the endocrine side last week, and the overall, just recapitulate what we saw, like going through the endocrine system was not, the idea was not to have you on each and every one of those 500 hormones that we thought that was important, but I think it was important that you understood that for many of the things that we're going to be talking about in the clinical cases, at least a certain amount of especially on the metabolic side, and also I think what I mentioned was on the metabolic and also on the growth, not the growth one, metabolic, and what was it you call them? But insulin, glucocorticoids in general, some of the stuff that you might need to control. We didn't go too much into the pressure side, but for example, minerals, acid, the acid balance, control. Those will be things that eventually you're going to have to come up with or to understand. I think it was pretty clear that for some of those things, you won't need necessarily a big knowledge of endocrinology, but hypothalamus, pituitary, and some of the major target organs. In that case, I think it was also not only the adrenal gland, but the kidneys, but also the lungs. Those would be very, very important for you to sort of understand. Today, I think what we're going to try to do is and I gave you a briefly introduction on the last class on the digestive system, and again, very bigger picture side of the control of the digestive system. Parts of the problems that we're going to we will address in the case studies will have to do, and I think the first thing that can come to mind is like, okay, so what happens if I stop eating? What could cause you stop eating or stop, better yet than stop eating, but if you're not able to get nutrients inside of your cells. That's the bigger picture when it comes to the digestive system. The other part would be, I mentioned that movement is a big component of the digestive system. You mentioned that you need to go and you need to break down, you need to digest, absorb, and from then on, probably generate energy from that. Part of the problems that occur with the digestive systems, or the digestive system in particular, is that if you, by some reason, have a problem with the movement of the nutrients inside of your gastrointestinal tract, the problem becomes that food, and this happens much more than in any other system, some problems may arise from that. And I think the first thing that we mentioned last class was like fermentation. So once you start having fermentation there, in some species that is desirable, you talk about ruminants, or you talk about some of the species that do what we call hindgut fermentation, such as horses and rabbits, you want some degree of fermentation. But the dynamics of that fermentation have to be controlled in a way that whatever the product of that fermentation can be utilized by the animal, and the rest can start to be either digested or you get rid of that. If you start, I think one of those cases, and we're going to go through some of these controls, intrinsic, extrinsic controls of the system, and to go in a little more detail, but I think the first thing that you have to imagine in a case is like, if your digestive system stopped working, stopped moving, what would be the consequences of it? before we mentioned fermentation. And then I think the next question over was like, what is fermentation? And what does it produce that can be bad for you? Okay. So we answer more or less what is fermentation and what are, I think one of the problems that one of you guys said in the last class was, for example, gases. So you start to have gases around and you have to get rid of those things. So this is one. Every time you start accumulating, for example, gas inside of your intestines especially, it causes pain. Yeah, you do have pain because of that. But the other one, which is probably most problematic, is fermentation. If fermentation is occurring, it means that bacteria is growing. They have to grow at a certain pace. Eventually, you can use some of the products that are inside of your digestive system, but eventually some of the substrates, for example, may run out. You don't have more to ferment. And eventually what happens is that those bacteria, they can cause not only what they produce, but even their death can cause lots of problems inside. So you start producing, or you start having accumulation of certain molecules or byproducts from the bacteria that can be damaging to the cell wall of the enterocytes, of the cells that compose the intestines, for example. And from then on, you can have all the way from damage to those cells, the death of those cells, and to exposure of those products that are inside of your digestive tract to the bloodstream. You can have even the bacteria going from your digestive system straight to your bloodstream. So let's start with some of these. We can basically skip this slide, but it just gives you like I won't ask you stuff on this slide specifically, but this is just the controls. You basically have controls that are based at nerval and endocrine secretions. Either from the outside of the stuff that is linked with your gastrointestinal tract to the things that are inside or tied to it. So you can have all the way, like some of these nerves, such as the vagus nerve, you're very familiar or might be very familiar with. It tends to control a lot of what's going on in what we call your splanchnic tissues. Splanchnic tissues is this technical term that defines everything that is associated with your gastrointestinal tract. And that goes from stomach, intestines, but also the pancreas, the liver, so the whole thing. Some of these hormones here, secretin, gastrin, cholecystokinin, GIP, and mutilin, those are all hormones that are produced basically once you start ingesting things. They can, make some of your glands produce juices for the digestion. Some of them will eventually stop once you start, for example, accumulating a lot of food in the stomach. That also produces a certain hormone that stops or inhibits ingestion. So it's almost like a self-control system for you to control your ingestion. And then you might be asking, so what controls exactly, for example, your thirst or your hunger? And those tends to be much higher level controls. When I say higher levels, to the level of the brain. The brain will be the one that will mostly control, and you'll have like, I think the way you can see like when you start, why you get thirsty, why you get hungry. These are, to begin with, these are very instinct kind of feelings. So this is the kind of stuff that is almost automatic to you. The body cannot, oh, I need to be hungry. No, these are very automatic things. It has to do with the level of energy that are circulating. You have quite a bit and obviously the hydration that you are. So the body has quite a few sensors around that measures all the way from how much salt you have in blood, which means how much water you might need, to the amount of, for example, of ATP or other reducing equivalents that you're consuming. Okay? So all of those things will eventually go into or communicate with your brain, especially your hypothalamus, the amygdala, which is mostly emotions, but they can have some effects as well. But basically, the hypothalamus will be the one controlling some of that. As I mentioned before, the wall, muscles, movement, transit time, this will be primary and very important functions of the digestive system. So to move the ingesta, to retain the food there, break up, circulate, all of these things will be extremely important. I think the other thing that we mentioned before about the digestive system is that, yes, it concentrates lots of your immune system, okay? Especially your innate, what we call innate immune system, which is this general protection that your body has. The rest, the next two slides are pretty. One is about before digestion begins, and also the esophagus. Like those are things that that are mostly obvious. Okay, so some of these problems, and in this case more than the other systems, like your heart doesn't stop beating, or your vessels don't stop dilating and contracting unless you have bigger problems with controlling that, which most of the time has to do more with humans than any other species. Like this stuff, for example, that we have lots of people with blood pressure problems, it has to do with your ability to dilate, and contract vessels. And this is a very human problem. Most of the other domestic animals that we deal with, they don't necessarily have that kind of problem because of their diet. Our diet is pretty bad, especially in terms of sodium. And once you do that to your body for 50 years, they lose the power to do certain things which are very important things. Your lungs, the same way. You don't stop breathing. If you do, you're not going to have a case to study. It's going to be just a past case. So, but in the digestive system, more than the others, you have this external problem, which is if you are not able to do this first step, which will basically end with swallowing, okay? All the way from the teeth to the mouth to the lips to the glutting. So this, for example, this is one of those very, very important systems where you have to be very good at differentiating what you breathe from what you eat. You start mixing the two, that's no good, especially when what you eat goes to where you breathe. That's a big, big deal. But these are mostly like mechanical problems. The same way that stops breathing normally has to do with a mechanical problem, which is muscles. For digestion, these first movements will be extremely important. Then once you go, obviously you have all of these things, lips, teeth, tongue, muscles, some of these nerves around your face and your throat, salivary glands. Those can be problematic, but those will be, relatively speaking, not always the case, but easier cases where you do know, at some point the animal is very old, lost all its teeth. The problem. But you know how to diagnose that, it's pretty simple. Sometimes the only thing is that sometimes you overlook those things. Sometimes it's a simple problem you think is, it's because of the diabetes and this. No, it has no teeth. That's it. But generally speaking, those are easy. The next one, and then you start the esophagus. The esophagus is the same thing. And the only thing that, I think it is worth, and it's probably this is going to be really, really similar to the rest of your digestive system, is that the movement of things in your digestive system that consists in this peristaltic movement, it's a very, very complex movement. Okay, so you have to have a very, very high synchrony of the muscles that are before the food and after the food. You have to basically go from a relaxation, passing the food, to a contraction. So you keep squeezing the food ahead. Okay? And sometimes what's going to happen, especially in the intestines, is that your food will go from ahead, and sometimes it goes backward a little. And it goes forward more, and it goes back a little. So you can have anti-peristaltic movements as well. But this is like this kind of movement, circular, longitudinal, and you have two layers of muscles around your digestive tract. They are extremely, like from a neural point of view, you know, how is it controlled? It's probably one of the most complex things you do. And that's why, especially as you get an older age, you start losing that, it's very difficult to recover. So you start having then some of those problems of having food that stays too long or don't stay long enough starts to happen as well. So on the digestive system, the first one, and this is the part that, you know, in terms of this, the organ, the stomach is the first one, and then it comes the intestines. The only thing that, you know, probably worth differentiating here is between ruminants and non-ruminants. I say this mostly because depending on the case that you get, I mentioned before, the ruminants, they are made to ferment. They're made to have like large amounts of feed inside of the rumen fermenting. Okay, that's part of what they do. And this idea that the idea that they can ferment something before it goes through their through stomach and also before it goes to their intestines for major absorption, it means that, you know, from a biochemical point of view, ruminants and non-ruminants are quite different. And what do I mean by biochemical point of view? It means that, you know, the way they process and they actually the molecules that they acquire for energy, they are slightly different. Okay? This case here, what do you think is going to be the major difference there between ruminants and non-ruminants? We're not going through details of the digestive. We're focusing on some major differences. Movement is very important. The controlling systems are pretty similar between the species. But this part of the fermentation ends up being important not only because of pathologies that may arise from not moving, but from a biochemical point of view between different classes of species, and in this case, because this is more of an animal class, you do have like bigger consequences of having fermentation occurring before or after actual absorption. So in this case, you do want a lot of this. This fermentation will have to be, they actually is, the principles of the fermentation occurring at a good level is exactly the same. You have to have a certain movement around the rumen. You have to have that amount of fermentation to occur in a very controlled way. And you have to have that thing moving on at a certain pace. Okay? So in this case, once you ferment, it basically means that you're using, let's say, a cow. Let's say we eat corn. Okay? So corn has lots of what? Starch. Okay? And once we eat corn, you have the starch. You can break that down. That's fine. And starch is basically what? Basically sugar. It's basically glucose. Is one glucose tied to the next. The same way that lots of other things, glycogen is also glucose tied to the next. Cellulose is glucose tied to the next. Cotton is glucose tied to the next. This stuff is one of those, what differentiates those is the linkage between those glucose molecules. Some of us, or most of the mammalian, starch, we're good. Cellulose, not good. Mammalians are that way. The only difference is that ruminants, they have bacteria to do that for them. And that's why they can use plant wall to acquire energy. So this thing means that, you know, for us, what we mostly absorb in non-ruminants, once we break down starch, we actually absorb glucose. We break it down, goes to the small intestine, goes to the small intestines, and we absorb. That's why sometimes, most of the time, after we get a meal, our glucose levels spike, goes really high. Once it goes really high, what comes next? What is the hormone that comes next? Insulin goes really high. Once the insulin goes really high, what happens to blood glucose? The levels goes down. So it goes to a more normal level. So that's what happens. On the opposite side, when you're hungry or you have like low glucose levels, what happens? What is the other hormone that is the opposite of the insulin in that sense? Glucagon. Glucagon goes, breaks down glycogen, and all of a sudden, glycogen breaks down into glucose, enters the circulation, your blood glucose level goes up. Okay, so this is very, very simple examples of eating a lot, not eating. Okay? In the case of ruminants, you have fermentation. During this fermentation process, you're using what for this bacteria to grow? Not only bacteria, but you have all the way from bacteria to other unicellular organisms there doing a lot of the fermentation. But what normally you use for energy of the bacteria? To provide energy for the bacteria. Think about making beer, a fermented product. What do you use as a base for beer? You can use everything basically, but let's say most of the beers.
Barley or grain?
Barley, wheat, rice, whatever whatever grain you have there, but use some sort of grain. Okay, and this grain has lots of what? Proteins and stuff, but it has, if you have barley or wheat, do you think they have sugar inside? Starch? They do, a lot. Okay, so you use that as your source of energy for the bacteria. So are you saying that, this bacteria are going to use a lot of that energy before it goes to the actual absorption from the ruminant? Yeah, it will. It's slightly less efficient, can be seen as that. Okay? But once you have that, you have to understand that at the end of the day, since you're utilizing the glucose, you don't absorb a lot of glucose. Ruminants do not absorb a lot of glucose. They absorb actually some other byproducts. It doesn't matter to us at this point what it is, but one of the things that happen to ruminants is that they never go there. For example, their blood glucose never goes as high as a monogastric, as a non-ruminant animal. Never goes as high. Why? Because they don't absorb that much, actually not much at all. That makes the control of these species, for example, that are ruminants, a lot simpler in terms of how they change or how you control glucose levels. They have very little to do with humans.
Why is it simpler? Because in like non-ruminants, if there's like no outside, like there's no like I mean, there are bacteria, but like, we don't control the bacteria, right? So it's just them doing the work. So why would it be simpler for ruminants?
No, it's not that it's simpler. It's simpler in the sense that it doesn't go up as much, doesn't go down as much. It's actually more complex, but in some ways. But it's just to have this, you see, it's a simple change in how you digest stuff. Both utilize energy well, both do okay. But just the fact that you have something before, you know, absorption makes a big difference. That's why in ruminants you can use weird feed stuff. You can use a lot of byproducts. You can use urea, for example, you know, to feed. And why? Because eventually you're trying to feed bacteria. You're not feeding the animal itself. You feed the bacteria first, and then the byproducts from the bacteria itself, or what they produce, is what feeds the ruminant. Okay. So the lots of this in terms of, you know, simple stomach, or even the ruminant stomach, the stomach, like 4 stomachs for the ruminant, contractions tends to be very important. You know, basically in one, especially on the ruminant side, you have to keep that fermentation chamber moving. You have to keep the movement of all the food that ingests moving in order for fermentation to happen well. In the case of these animals that are, you know, non-ruminants, in lots of these, you actually need the contraction of the stomach to help actually physically decrease the size of particles. So you need your stomach to be pretty powerful. I mentioned before, so super environment for fermentation. You do have, and you need, you know, to feed bacteria. And 4 compartments. In general, ruminants have 4 stomachs. But these things here, contraction, buffer, and retention, these will be very important. It will be important for non-ruminants. But for ruminants, they tend to be more. One of the things here, nice, and you're going to see, since it's buffer, and we love buffers in this class, is the idea that, you know, you do need buffers in this case, because a lot of what you produce during fermentation is like not exactly powerful acids, weak acids, but in a very, very high amount. So in general, every time you produce, or every time you're fermenting food, the pH of that environment goes up or down. Goes down. You're reducing some of these acids. Some of these acids will have to leave the stomach as it goes to the intestines. Some of those acids are actually absorbed into the rumen, not actually stomach, but you have to control that. Now, this is a very nice, for example, very, very simple case study. If you give something that is highly fermentable to an animal, such as a cow, you don't want to give too much of it. Why? Because it ferments and you drop pH too much, you start having problems, not only with the population of bacteria that is in there, but actually to the absorption and even the wall, the rumen wall or the intestinal wall after that. They just, if you start going, your pH start going too much down, you start having or start damaging those cells. So ruminants have a weird, I shouldn't say weird, but they are much more sensitive to some of those things. Okay, so I think the idea here is that you understand, well, in ruminants, you're feeding bugs first, then you feed the animal. In all the other ones, you're feeding the animal first, and then eventually you will control population bacteria as well, the digestive tract, but it's a different dynamic. Does that make sense? Yeah? So some of these things in small intestine will be by far the largest site of absorption not only digestion, but absorption. And I mentioned this motility pattern as digestive and interdigestive phase, is that this digestive phase, I think I might have said this in the previous class. Once the food goes into the small intestine, it's already pretty much broken down physically a lot. Some of those things have already started to become digested because of all the juices that are in the stomach. And then it goes to the small intestine. The movement there was always like this. They go in pockets. So, and you have to have like it digests. Sometimes it breaks down more. Sometimes, no, most of the time it breaks down more, and then it moves a bit more, and then it absorbs, then it digests a little more, and then absorbs. But they do it like in little pockets. They need some time for things to happen. This is what we call the digestive phase. And then you have the non-digestive phase, which I mentioned before, which was this, and we call this movement MMC, or migrating motility complex movement, which is like this strong, continuous movement or contraction of the small intestines to basically the big cleanup. Why? Because you need to get rid of whatever is left in there. You don't need, or you don't want any remaining food during the digestive phases or in between the digestive phases left over inside of the intestines, okay? So you end up controlling bacterial population, you push undigested material as well. And this is, believe it or not, but this has a big component into, or a big importance. Do you remember when the biome starts to become like a hot topic? So what kind of bacteria you have inside of you matters more than anything, and so on. A lot has to do with that type of movement. Of course, it's not the only thing, but you can imagine, like one of the things that, for example, people have done quite a bit of research in animals, trying to come up with, what is the microbiome of the gut of some of these animals, and how is that related to, you name it, growth and production of things and so on. The problem becomes exactly because of this high turnover of things inside of the gut. Probably your bacterial population at noon, very different than your bacterial population around 5 or 6 in the afternoon. That is different than what it is. So obviously at some point you probably have a certain cycle, but it's more complex than just saying, I'm gonna see and I'm gonna get what is the population of the bacteria inside of me. And that tends to bring actually more questions for research than answers. So the colon is, and when I say the colon, and we can say here like the large intestines as well, it has a lot to do with water and electrolyte absorption. Okay, so this is the phase where a lot of the food that you ingest contains a lot of water. So what they try to do is that in the beginning, you actually need quite a bit of moisture for breaking down and absorption to occur. But once that is done, eventually goes to your hindgut, and you have a lot of water being absorbed. So if you do have quite a bit of problems in your large intestines, in general, it means that you're going to be absorbing water very poorly, okay? And you do have yes, in some species you have fermentation, but in this case, the fermentation will occur after the major digestion and absorption has occurred, which basically means the rate and the amount of fermentation that will occur doesn't even compare with what happens to ruminants, but it's still very important. Okay, it's still quite important. This ileocecal, that's okay. I won't go too much into that, but it has to do or tied up with the movement side that I was talking. After it gets to the cecum, which is one portion of the large intestine, they have a very strong sphincter there. When I say sphincter, it's once it passes through, it's very hard to have anti-peristaltic movement going backwards. Why? Because at that point, the body understands that, I have absorbed most of the nutrients that I would like at this point, and then the rest, it will be mostly water and some other things that I might absorb, but you don't want to mix any more things going backwards, okay? I think this is the part where I mentioned this during this other, I'll be right there with you. This non-fermentation, digestion, absorption, I think we went through this, in terms of, what is absorbed and what is actually, what is digested and what is actually absorbed in fermenting, in ruminants and non-ruminants, depending on the species, of course. Physical reduction, I think this is a given. Some of these other things in terms of, what happens, and this is more of a curiosity here. Most of the breaking down of food that happens in your body is classified into this bigger, class of chemical reaction that we call hydrolysis. It doesn't really, you don't need to know these things, but, when you talk about digestion is not only breaking physically down a certain food, but you're going to have to break a few linkages. Do you have to break everything? Of course not. Okay? So you never want to break down glucose inside the lumen of the things. What you want to break is just the linkages between several glucose that are together. Okay? And this goes on also for fatty acids. So if you think about triglycerides, what you want to do is try to separate what composes a triglyceride, which is one molecule of glycerol with 3 fatty acids. So do you want to break down fatty acids inside of the lumen? Of course not. You want to leave that to inside the cell where it can generate energy. But you want to break down this bigger package into smaller ones. That happens also to amino acids, and that also happens to protein. Protein turning into amino acids, triglycerides, and some other fats into smaller components. And obviously, on the sugars, those sugars that are polysaccharides, for example, to go into monosaccharides or disaccharides. So you want to break this bigger linkages, not everything. Some of these things, I think this portion here you have probably heard before. You have a luminal phase of digestion and a membranous phase of digestion. Overall speaking, what happens is that a lot of the digestion occurs when you mix a lot of stuff of these juices, pancreatic juices and so on, inside of the lumen. But eventually, when it gets closer to the membrane, to the enterocytes, really close to the cells, you have a little bit more digestion because that's the fine-tuning until it gets actually absorbed. You need to understand that, the enterocytes, they have like an enormous surface because of the villi, that they are inside of the intestines. And they have to absorb, if you really think about it, an enormous amount of material. Okay, so they have to be quite fast in doing that. So they try to do a big phase first inside the lumen, and as you move on a little bit, and you have more of that juice really impregnating the walls of the intestines, then you start having more of this membrane digestion and so on. Is that really, really important for some of the cases that you're going to see? Not really. Okay, this is more, physiology, knowledge on it. Yes, I think we mentioned this, bacteria, microorganism action for fermentation, fermentative digestion. We mentioned that, it's going to happen mostly because the major reason for fermentation to occur is to break down stuff that we can't. But during the process, yes, the bacteria utilize stuff that we would, such as glucose. Okay? But then the organisms or the mammalians, they evolved in a certain way to use also these other, these other byproducts. It sounds very weird, but a lot of the nutrition from ruminants is actually the bugs themselves. So you have lots of this bacteria growing. What do you think happens to that bacteria? It goes to the small intestine. And what happens in the small intestine? Yeah, they become food. You know, and this is one of the things that is very weird about the ruminants is that, yes, they utilize the actual bacteria as a big portion of their food. Yeah, sometimes you think, oh, that is gross. No, it's not. They're very efficient. With that, all these things, pH, moisture, ionic strength, oxidation reduction, all of this like biochemical stuff will start to become very, very important, particularly for fermentation. You're going to have to control, or you're able to control a lot more than non-ruminants. And obviously, you can have either what we call four stomachs or HIMCA. These are the two classifications, official classifications that we have from fermenters, digestive, fermentative digestion. I think this part here, I think it has more to do with again the movement of stuff. You have to have a certain flow of things going. Generally speaking, not only for ruminants, but too fast, terrible. Generally leads to diarrhea. Why? Because you cannot absorb water as well. Slow is also pretty bad. You ferment gases, damage to the wall. Has to do with some of that. So the overall speaking, I think the message for the digestive side is that you understand some of these basic anatomical things that are attached to the gastrointestinal tract. Pancreas, liver, of course, will be the two major organs that will be controlling some of the functions there. Not only because they, in terms of the pancreas, of course, it is the glucose control. The liver will have a pretty big or a pretty large effect in, for example, digestion of fats. But they also, I think the liver is especially important afterwards. And that's where the biochemistry on the digestive side comes in. That's why I emphasize a little bit the fermentation example, because once you start digesting different things, your biochemical pathway on how you absorb and how you generate energy becomes very different. Okay. The next slide is probably has a lot to do with give it a quick read on some biochemistry book, or I think it goes back again, as we were mentioning, acid-base balance. Now for the generation, for example, of nutrients, and glucose is one that it becomes very important. But the idea that the more you know about some of this basic stuff, the easier it will be for you to understand the case studies, or during the case studies, you eventually will have to go back to those things. And that's why we're going relatively fast into this to go. And then I think the next one, what I would like to do is actually go to some of those case studies. So the glycolysis, gluconeogenesis, transport system, amino acid, nitrogen cycle. I'll never cover those things individually in the tests, in the quizzes, or either the final. But very likely that in one or the other case study, you have to go back to this a little bit, grab information, try to go into this. Again, on the endocrine, not tying these with the endocrine class, I think it is when they talk about the metabolic hormones and so on, it will make your life easier. It truly will. Very good. So start. Very good. Let's get a little break right now, and then we will return. Yeah, I think you had a question, and I didn't give you time to.
It's the sphincter that controls it for nothing to go back.
Yeah.
If there's a problem with digestion, does it allow things to go back? Or
I think there's a certain point inside of the, for example, it's very common to have from the stomach back to the esophagus as vomiting can be induced, or stuff that, for example, if you eat something that is is perceived as dangerous or toxic, and normally the body has ways to make that. Inside the small intestine especially, you have, as you said, like peristaltic, a little bit of antiperistalsis, and go back and forth. And even in the large intestines, you can have, like in the cecum, there's lots of movement of things. But eventually when they leave the small intestine to the large intestine, they go back. And if you do, then you start having actually issues with that. Yeah. Okay? let's go this next 20 minutes. And the idea why I try to go in this first basically 2 weeks, going for a third week to finalize all the way to renal. Probably we'll take a portion of the next class also to study the kidneys. Is that we can finish off parts of very briefly on the circulatory, respiratory, digestive, renal, yeah, some of the endocrinology part, but at least we have a big deal of what we consider the homeostatic or homeostatic systems covered. And then I think the next class we're going to have oh, by the way, we have quiz on Thursday, and let's cover, I think we're going to cover mostly, I think it's just the very initial homeostasis portion of the first class, and then cardiorespiratory. Okay, so there's homeostasis and cardiorespiratory. So the idea for the kidneys then is to finalize this and especially try to, this will close the loop with how we control pressure and also how we control acid-base balance. So with these two things, pressure, acid-base balance, and then we have ventilation, very basics of endocrine and digestive, we're going to be enough to start, after the quiz, we're going to go through at least a couple of case studies where we can, I think the idea now is that we exercise that knowledge, and then we go through some of this very simple, some of these are very simple cases that we can think about it and come up with ideas. I'll try to shut up more, and I'll make you guys talk more a little bit. Is that okay? Very good. So, is maintaining homeostasis, but what do you mean by this? What do you mean by the kidneys trying to maintain homeostasis? So, from the top of your head, what is the major function of the kidneys? Oh, it's to filtrate the blood. I was reading to my daughter the other day, these are the kidneys, and that filtrates the blood, and that's why you pee. Okay, excellent. Go a little more technical. What is that exactly the kidneys are filtrating? And what is that they are trying to maintain? Like some of those things we have already talked, through with the other systems, But give me one example. Like we're going through all this, but
they try and maintain water and solute levels.
Yeah, so you're trying to maintain water levels at a certain level. Okay, so if you're not drinking a lot of water, your kidneys will try to retain water. If you're drinking a lot of water, or if you're drinking a lot of alcohol, your kidneys, because they want to, or because it is just an effect of alcohol, you're going to pee a lot. Okay? So the idea then is that, the kidneys have a very, very, complex but very effective way of concentrating or diluting your urine. Okay? But Ronaldo, wouldn't be like if they can concentrate all that much, wouldn't it be nice to just drink less water and the body can just retain more? It could, the body could, but why then it is good that you drink a lot of water? Not like 10 liters of water, but why is it good that you drink a lot of water? Why do we tell our kids, oh yeah, it's very clear the pee, so it's good. When it's too amber color, they say, oh, you should be drinking more water. Why do we do that?
Because the water helps to get the waste out.
Yeah, so it helps to dilute the waste, and it helps to actually generate more waste. Once you have more water, and this water will be very important for you to actually move metabolic processes inside the cell, it's always good that you have more circulating and almost like flushing the whole system more often. Even more important than that, then you're going to start metabolic or excrete metabolic waste. This will be important, especially, obviously, from a physiological point of view, you're going to, almost all the time that we're talking about urine, you're talking about urea, and you want to get rid of the urea. The other one that will be super important is that once you start talking about drugs and how we metabolize and excrete drugs, antibiotics, anti-inflammatories, and so on, this becomes important because that's normally the way, not all the time, but either the liver or the kidneys, sometimes the lungs will be the 2 or 3 major ways in which you're going to excrete stuff. So depending on how aggressive a certain drug is to a certain organ, you may choose a certain treatment or another. So if you're already a certain, or you do have already, an animal has a certain immunodeficient or kidney deficiency, renal deficiency, you may want to choose a drug that is less aggressive to the kidneys because they can already not do their job very well, so you try to minimize that. So some of those things that, it does, so the other part is that, some of these low glucose and low molecular weight proteins, this is the stuff that you're trying to, a lot of times, retain, okay? You don't want to get rid of useful proteins, you don't want to get rid of glucose, and obviously, you're trying to control water and all this. All of this, you can already imagine that all of this, but especially the water, is going to affect pressure. Okay? By the time you start not having too much water around, your blood pressure will necessarily go down. Okay? Which then, you see, if your kidneys are not working well, it's very likely that your heart will suffer. If your kidneys are not doing well, your vessels will have problems. And you're going to see that if your kidneys are not working well, your acid base and your lungs again will suffer with that. You see, this is the part where you tie a lot of these things. So they respond to water, as I said. You mentioned well, electrolyte, acid base disturbances. You're going to see that the electrolytes, in this case, sodium is by far the most important, which will be tied up with water. So you can almost say that, the water and the major electrolyte here, sodium, will be tied together, on how it is controlled. If you reduce the amount of water, generally speaking, immediately you increase the amount of salt or sodium, and that triggers you retaining more water that can keep the sodium low. And of all the hormones that it is produced, renin is the most important for us that we're going to discuss in this class. EPO, or erythropoietin, will be also important, but is a hormone that is produced in the kidneys, but will have a major effect into the bone marrow for the production of red blood cells. Okay, so we made them. We'll go into that, but it's not the major thing that we'll be talking about. Okay. Functional unit. Now go back to the drawers of your brain and try to recollect what you know about the nephron. We're going to discuss this again, but basically speaking is the filtration unit of the kidney. Okay, so as the filtration unit, you can imagine that, we'll have, by filtration, it means that, something have to pass through a certain membrane or a certain barrier, and eventually after that will have to be reabsorbed. And you can control this reabsorption. That's how the nephron works in terms of, the controlling of glucose, water, and low molecular proteins. So the overall speaking, this this is like a cross section of the kidney. Kidney looks really like this inside. It has like this cortex, followed by this kind of, medullary like components that are tied to a duct system. And from this duct system, obviously, it goes to the ureter, then to the urethra, or to the bladder, and then urethra, and that's where by the time it goes to that white portion there, it's already the urine. And from there, what you try to do is to either store and go in the bladder or the urinary bladder, and then basically go to the external environment. The kidneys then, as opposed to obviously the digestive system has nothing to do with movement. You're not supposed to have access or contact with bacteria, fermentation, blah, blah, blah. That does not exist. It cannot happen. But this is much more a biochemical kind of, is much more sensitive to biochemical changes in the body. What you have here is that each of those things here are what we call the nephron, that is tied to a tubule, that then is tied to another bigger, set of tubes that are actually tubes that actually go to the ureter. The blood, you can imagine, like these are basically the tubules that makes up the nephron. Around these tubes, and imagine that you have like thousands, millions of those units tied to packed together. And around each of these, you have a little vessel going in. So the idea then is that the blood that runs through your body will have to run to your kidneys several times a day. So if you add up the amount of filtration that your kidney does in the day, and let alone in your lifetime, is enormous. I mean, enormous. Almost all the blood that you have will have to go through this membrane here. All this stuff goes through here. And when you say, what do you mean by all this stuff? This stuff is your blood, your red blood cells, your white blood cells. They don't cross this membrane. When I say the membrane, this Bowman's capsule? There is like very, and this is the glomerulus. This is the vessel portion. This is the kidney portion. In between this, in this space here is where the filtration happens. By filtration, meaning water and some of the very low molecular weight, molecules can pass through. Okay? The other things, such as cells, very large proteins, such as albumin, for example, cannot pass through that. So what you do have is that it's almost like the blood comes this way, almost gets like dehydrated to this. The blood that comes out this way is almost like very kind of concentrated. But then the amount of stuff that goes through this and is actually, so all this water comes to this and is rapidly, leaves the tube very fast. Okay? Why? Because you control that thing, and what you're trying to do is to concentrate a little more what you would like to get, not to get rid of it. But water in general passes through this, but all of a sudden it dissolves back to the interstitial space. And from the interstitial space, and when I say interstitial space, all this white space, we do consider interstitial, and then it's picked up by this vessel again. So it's almost like it dehydrates, hydrates. It dehydrates, hydrates pretty fast. However, inside of that tubule, you still have this stuff that normally speaking, you'd like to check first with the body. Do I keep it to go to the urine, or do I let it go into the interstitial space for reabsorption? Okay, so this is how the kidney, overall speaking, works. The only thing is that around this area here, you're going to have lots of sensors, especially particularly in this area here, you know, in between this like distal tubule and the Bauman's capsule. This is an area that has lots of sensors, and we call them the juxtaglomerular complex. And this is an area which has sensors that, for example, can tell if there's too much sodium around or too little sodium around. And this is normally the space where renin can be synthesized. Okay? So the next slides have lots of, you know, capillaries that extrude the fluid, plasma composition. So when I say the plasma composition here is that, you know, once the water comes to these tubules here, whatever is here doesn't have red blood cells, so it's very light in color. It sounds more like the plasma portion of the blood than anything else. Okay? What do you call, oh yeah, the other thing that sometimes in the slides they go, once he enters this part, this plasma-like portion, you call this the filtrate, the glomerular filtrate. And then this filtrate will go through these tubules. Either it will be retained or be removed from the tubule to the interstitial space, and then it goes. Go ahead.
What's corroding that bond when there's things that are also feeding the urine, like glucose or protein?
So very good. That's a good question. So the question is basically what's going on when you start having stuff that goes all the way here that shouldn't be there, such as glucose. Okay, so glucose, you're supposed to have, overall speaking, not much, first of all, not much going through this. It's a relatively large molecule, okay, so it's not supposed to be around, but you do have. Okay, you do have quite a bit of it. The problem is, at some point, you're going to have some of this glucose going in this direction. There's two things that can happen on the glucose side. One is this reabsorption mechanism doesn't work, or one, you can have damage here. Sometimes if you have damage here, you start piling up like large molecules going in, including proteins and other large, you know, substances, and then you end up having, there's not enough time for you to reabsorb or to get rid of to the inside of the tubes, to the interstitial space. So there are conditions where you can have like what we call glycosuria, which is everything that ends up with urea in clinical cases is because it is present in the urine. So glycosuria is basically glucose in urine. So you have some types, for example, of diabetes that you can have quite a bit. And one of the reasons why you end up having quite a bit is that you actually have too much in the blood. And as a lot of the diabetic people, they have hyperglycemia. One of the ways they find is to get rid of the glucose through the kidneys. But then it's because of mass amount of glucose going through the kidneys, which in the long term can cause damage to the kidneys itself. Okay? And this is, for example, the reason why they, people with diabetes, for example, they have large volumes of urine. You know, they pee a lot. And one of the reasons is that because they have too much glucose coming in here, that glucose creates hydrostatic pressure to the inside the tubule too strong. So they actually get water from the interstitial space and sucks to the inside of the tubule, and you end up creating a lot of diluted urine. Although it's diluted, it still has lots of glucose. That makes sense? That's
So with like bigger molecules, if they get through, would it be possible for them to like get stuck in a way? so small? Or?
Again, because they're small.
Because like the path is small.
Oh, the path, you mean this?
Yeah, I'm just wondering if like
No,
that's how.
the tubule compared to a molecule is very big.
Okay.
Okay, so it's not like this is, obviously this is very amplified, but this is it's a very long tube and molecular, you know, any molecule will be a lot smaller than it. I think these fenestrations here that allow whatever it is to pass through from the glomerulus to the Bowman's capsule, this is the one that, you know, can be bigger or smaller. And lots of things actually do affect how things are filtrated. That goes all the way from the size of the molecule to the charge of the molecule. So if you have molecules that are, for example, negatively charged, they tend to be more difficult to pass through. This is just basically cellular biology there. And shape. Sometimes the shape of the molecule also can, if it is too long, they have a certain, sometimes it's more difficult. If it's more like rounded and so on, it tends to be easier. So there's other things that can, I think the charge is the one that affects the most. Size and charge is the one that affects the most, but the other ones. So theoretically, you know, even large proteins, some will pass. You know, it's not like it's 100%. Cells, that's a problem. You know, by the time you start having urine, and so a typical case would be if you start seeing red blood cells in the urine, a problem. Okay. So I think we're done. Okay, so we'll start from here. So this is just a last example here is the filtration, the filtration rate, which is, if you think about it, per day, you're going to basically have from a beagle, from a small, relatively small dog, the filtrate, you know, the one that comes from the glomerulus to the inside of the Bowman's capsule, you have almost like over 50 liters of filtrate going through that. So which means a bagel will, how much urine a bagel will produce a day? Not even a liter. So which basically means the whole 53, 52 liters there will have to go back to the blood, to the vessels. So it's a very, filtrates everything, but basically returns almost everything. But during this process, you have that very, very small amount of time, but also very important period of time where the liquid is inside the tubule where you can retain or let it go. Then the body makes it. Okay. So by the time we finish off this, you know, the last class, we should be able to finalize all the stuff on the blood pressure. And then we can see at least 2 cases there to play with. Is that okay? Very good. Thank you very much. See you.